Okay, well, I'd really like to welcome you all to this breakout session. My name is Daniel Peckham. I'm a clinical academic in Leeds with a particular interest in CF, the gut and inflammation. And this session is going to really focus on gastrointestinal tract. And we've got 4 young dynamic speakers. And the first speaker is Dr. Jennifer Caley, who's going to tell us about the impact of restoring luminal fluidity on gut microbiota and gut health in CF. So welcome, Jennifer, and let's start.
Okay, thanks very much. I'll just share my screen. Okay. So my name's Jennifer Caley. I'm a postdoctoral researcher at the University of Liverpool. And in this short talk, I'm going to give a background of this SRC, talk about our progress and next steps, and also how this SRC could potentially improve the lives of people with CF. So the CF gut is an extremely dehydrated environment with sticky mucus accumulation. This leads to frequent gut problems such as constipation and DIOS. It's thought that this altered luminal environment also changes the normal bacteria present in the gut to a more dysbiotic state, which could be contributing towards disease development. So improving the hydration, or what is known as the luminal fluidity of the gut, may alleviate some of these gut issues experienced by people with CF. And this was the issue that this SRC tackled. So our approach was not to influence CFTR function, but targeted another protein called NHE3, which also functions abnormally in the CF gut, but is not mutated like CFTR and therefore provides more potential for manipulation or modification. So in the CF gut, NHE3 is hyperactive, and this causes excessive sodium and fluid absorption and subsequently a dehydrated gut. So initially, this SRC aimed to understand how the gut microbiota of the small intestine are altered in people and mice with CF, as this had not been looked before in humans in this particular area of the gut, as well as understanding how the host gut physiology is altered. So importantly, in this project, we used mucosal tissue to study the gut microbiome, not fecal samples, which are more commonly used. And we did this so we could characterize the adherent mucosal attached bacteria, which have a bigger influence on host health than the transient bacteria present in the luminal content and the feces. So then the SRC looked at whether an already FDA-approved drug targeting the protein NHE3 could improve luminal fluidity and gut symptoms. And this was carried out using CF mice and non-animal models known as organoids or mini-guts. And then finally, we looked at whether treatment with these hydrating drugs could resolve gut symptoms such as blockages and whether they influenced the microbiome and host epithelial cell composition to try and understand how this may impact host health. So we're on the last year of this SRC, and we've made good progress in addressing all of the aims that we set out to at the beginning of the project. We still have quite a bit of work to do to complete. So we've produced a number of publications, some of them are shown in the slide, and presented work at numerous conferences and meetings. So some of the aims that we accomplished include full characterization of the dysbiotic microbiome in the small intestine of CF mice, and also people with CF using mucosal tissue. So this work revealed that the gut microbiome in CF has a much higher abundance of pro-inflammatory pathogens and bacteria that are adapted to survive in this altered luminal environment. So we also showed that in humans, there was a huge abundance of bacteria often found in the CF lung, suggesting there's some link between the microbiome of the CF lung and gut, although a lot more research needs to be done in this area. So we also demonstrated that the intestinal epithelial cell composition is completely deregulated in CF mice and humans. And as the gut microbiome plays a big role in controlling this process, it's possible that the microbial dysbiosis observed is linked to these host epithelial cell changes. So colleagues in the SRC also did a huge amount of work testing an NHE3 inhibiting drug in CF mice and also on CF organoids, and revealed that it reduced fluid absorption and increased alkalinization, suggesting it may increase luminal fluidity. So us and colleagues are currently preparing publications answering the final 2 questions of, does the NHE3 inhibiting drug prevent intestinal blockage formation and reduce inflammation, and does it influence gut microbiome composition? So this project's been a real multidisciplinary effort. It's allowed massive knowledge sharing across specialities and also the sharing of specialized techniques. So, for example, one of the co-investigators is an expert in the creation of these organoids or mini guts, and currently there are plans in place to teach this technique to another co-investigator for use in a future project. So it's also led to the formation of new collaborations between the co-investigators who've submitted grant applications for future projects using these shared techniques. So the next steps in this project include the publication of the final few papers, which are in preparation now and should be submitted and published very soon. So this project has huge potential to influence the lives of people with CF in the future. So firstly, by demonstrating that the NHE3 inhibiting drug can influence fluid absorption and luminal fluidity, there is potential that this could help gut symptoms in people with CF. So it's hoped that this project will set the groundwork for future clinical trials in people with CF. Secondly, this project identified that the gut of people with CF has a high abundance of a bacteria which is sometimes found in the lung, suggesting a link between the gut and the lung microbiome. And this provides an opportunity to possibly influence lung symptoms by manipulating the gut microbiome. However, this is just kind of the first step in this area, and a lot more work needs to be done. So we also found that the gut epithelial cell composition of mice and people with CF was extremely abnormal, with a possible link to the changes in the gut microbiome. So finally, I'd just like to thank all the collaborators that were part of this project, and obviously the CF Trust for funding it.
Well, thank you really very much, Jennifer. I think that's really fantastic translational research, and you can really see the long-term impact. I'm going to move on swiftly to the next presenter, which is Laura Caley, who's going to talk talks entitled "What About My Tummy?"
I'll just get hopefully you can see the slides. So hi, I'm Laura. I'm a dietitian completing my PhD, and I'm delighted to be giving a brief overview on my research regarding SRC number 12. And we're investigating the gut microbiota and links with inflammation and lung health. The bacteria, which make up the majority of microorganisms in the gut, are often referred to as the gut microbiota. And the diversity and types of bacteria can vary widely between people and diseases, and they can communicate with and modify the immune system and alter inflammation, both locally and systemically. Further, the different types and balance of bacteria have the potential to increase or dampen inflammation. We are also investigating the relationship between the gut microbiota, diet, abdominal symptoms, medications, and how these all change with new CFTR modulators. I'll be focusing on our observational cohort study, which was in pancreatic insufficient adults with CF across 4 UK CF care centres. Here are our preliminary findings. In this slide, I present data on abdominal symptoms measured with the CF Abdo score, which was developed by our collaborators in Brandenburg University in Germany. And we wanted to know whether CF-related diabetes exacerbated GI symptoms. And this is because several studies have found an increase in specific GI symptoms in both type 1 and type 2 diabetes. So I have 2 bits of data here. So the first on the left is comparing abdominal symptoms in people with CF without CF-related diabetes in the green to those with CF-related diabetes in the orange, with a bigger area of shading indicating a higher burden of GI symptoms. And in summary, people with CF-related diabetes did have a higher burden of GI symptoms, including more acid reflux, bloating, and nausea. On the left, we have some very preliminary findings on abdominal symptom changes with triple CFTR modulator therapy. While some symptoms improved, at follow-up, almost 80% had fatty stools and half abdominal pain. And we're investigating this further to better understand the underlying etiology and how best to manage these troublesome symptoms. In collaboration with Professor Shortchain, we're also shortly to publish on a larger study regarding changes in GI symptoms with triple therapy. Next diet. Again, I've got 2 bits of data here. The first on the left is looking at nutritional intake in adults with CF, not on CFTR modulators, and looking at different groups. And the key findings were that females tended to have a lower energy intake than males, and those with a fast rate of lung function decline tended to have a lower energy intake than those with a slow rate of lung function decline. Also, energy and protein intakes didn't directly correlate with BMI, which just shows how complex diet and nutrition is in people with CF. And then as a taster, on the right, we've just started analysing changes with CAFA trio in people who were previously CFTR modulator naive. And unsurprisingly, BMI increased, but energy intake actually decreased, which suggests the weight gain is not simply a case of eating more. We're conducting in vitro studies as part of a parallel research program, which we feel may help uncover the potential reasons for this weight gain. Lastly, the gut microbiota. We're awaiting results from the Sanger, but what I can show you is some parallel research we've done at the University of Leeds, comparing the gut microbiota in people with CF to those with colorectal cancer and healthy controls. And this graph is comparing differences in gut microbiotal composition between individuals. So the closer the dots, the more similar those two people's gut microbiotas are. And the orange dots denotes the samples. And as you can see, they cluster distinctly from both the colorectal samples in the red and the healthy controls in the blue. And we're investigating this further and looking at the specific bugs and toxins because we feel this is an increasingly important area. So in summary, we're awaiting the in-depth gut microbiota profiles, and we'll link that with diet and abdominal symptoms and how these change with new CFTR modulators. Secondly, we will further investigate the gut microbiota and toxins in people with CF compared to those with colorectal cancer. And thirdly, we'll analyse stored blood samples to identify potential biomarkers of inflammation driven by the gut microbiota. I hope you've enjoyed this brief overview, and we look forward to sharing a lot more in the months to come. I would like to thank my supervisor, Daniel Peckham, the rest of the SRC, and of course, all the study participants and you all for listening. Thank you.
Well, thank you very much, Laura, for that fantastic summary. We're going to move on because I want to keep to time, and I'm going to invite Ryan now to come and give his presentation, which is the organisation and function of the gut microbiota in CF. Thank you, Ryan.
Okay, thank you, Daniel. So I'm Ryan from the Department of Life Sciences at MMU, and I'm going to dive into the organisation and function of the gut microbiota in CF. So first of all, why should we care about the gut microbiota in CF? Well, we know in CF there are a wide array of GI-associated problems, such as the site of the intestines, the pancreas, the liver. So it's therefore unsurprising that there are also an array of persistent GI symptoms, of which we know is a top research priority to understand and alleviate such symptoms. If we look at the site of the intestinal tract itself, we know there are intestinal blockages, inflammation, lesions, and increased risk of malignancy. And we actually know that there's evidence to suggest that there are relationships with the dysbiosis of the microbiota with such of these issues. If we step away from CF, we know that the microbiota as a whole is pivotal in modulating and impacting host health through the metabolites that it produces, such as short-chain fatty acids, bile acid modifications, and also in immune tolerance and immune learning in younger patients and people. So if we dive into what we're looking at, our update so far can be split into two chunks. So the first side is a cross-sectional analysis between healthy controls and people with cystic fibrosis, where we wanted to know which clinical factors associated with the composition and the differences between the two groups. And what we initially found was that actually antibiotic usage, pulmonary antibiotic usage, alongside the basic CFTR defect, really did drive the differences between the two groups. On top of this, we also found that intestinal physiology and transit also significantly contributed to the growth. And this was done through our collaboration with Nottingham. More specifically, in terms of the intestinal physiology, it was things like oral-fecal transit time and small bowel water content, which really expanded this community of rarer bugs or bacteria, which were able to expand in the CF population. So moving on, we're now looking at CFTR modulation, in particular Symkevi administration at the minute. We're just tying that up. And again, similar principles are being investigated as in the previous work. Also incorporating abdominal symptom scoring and inflammatory markers. What we can show you so far from the preliminary data is that actually the effects of Synkevi aren't too profound in terms of reverting the microbiota back to a state of a healthy signature, if you like. And that's really depicted by this cluster of blue points on this MDMS plot, which basically shows that the healthy control samples from our first study, of which the matched CF patients went on to Synkevi and placebo, are very distinctly clustered away from those that do take Synkevi in the CF group. So obviously we want to understand why this is happening. Is there any overlying or arching factors that are stopping this from happening? But we also want to understand the functionality. Is the composition different, but is the functionality the same? And how can we do that? Well, we can do that predictively by looking at sort of incorporating long read sequencing approaches, where we can then test for or predict metabolic capability, antimicrobial resistance genes, virulence factors. But then we can also directly quantify and measure this by integrating chemistry techniques like NMR spectroscopy or GCMS. So for example, to quantify the levels of short-chain fatty acids in a fecal sample and see how that ties into the bacterial community that we're observing, and to see if there's like elements of redundancy. So in CF, does a certain bug step up and fulfill the role of a different bug that would be more prevalent in health controls, and so on and so forth. Obviously, we also want to look at the effects of Kaftrio. So does Kaftrio administration directly or indirectly impact the composition? Does it do a better job than Synkevi at reverting this microbiota back to a signature more similar to healthy controls? Are there any relationships between gut function, composition, and its usage? But also, again, touching on previous points from the presentations, is there a gut-lung axis sort of interaction happening? What's the level of bidirectionality? Is there metabolites produced in CF gut that impacts health in the respiratory domain, and vice versa upon Kaftrio usage? And then finally, just to touch on inter-kingdom crosstalk in the CF gut. In the CF airways, we know quite a bit about fungi now, but again, in the CF gut, this is widely overlooked, and we don't actually know the level of interaction between fungal and bacterial communities. And there is evidence to suggest that there may be interactions in other similar inflammatory areas or diseases. So following on from this, opportunities for future collaboration would include an expansion of the current pilot studies that I've just mentioned previously, to again touch on these underlying themes. So things like antibiotic usage, really diving down into class-wise analysis, and obviously having a larger cohort will make us make that much more easier and give us more power for statistical analysis. Of course, collaborations with CF centres focused around PPI is highly desirable to make sure that research aims are realistic, considerate, and attainable. And finally, the incorporation of sophisticated top-down, untargeted metabolomic approaches, which essentially means more sophisticated ways to find novel compounds that may be in the CF gut that could be contributing to host health. So I'll leave it there. Thank you for listening. And yeah, thanks to my supervisory team and all the sponsors.
Well, thank you very much, Ryan. It's really fascinating seeing how all these projects that are funded by the CF Trust or co-funded are so integrated. Our last speaker is Alex, who's going to update us on clinical gift studies. So Alex.
Hi, everyone. Thank you very much for the introduction. I'll just share my slides. Okay, so good afternoon. My name is Alex Yule. I'm a clinical research fellow and paediatric registrar from the University of Nottingham. I'd like to thank the organisers of the UK CFC for giving me the opportunity to give you this presentation. We'll aim to give you an update on the gut imaging for function and transit in cystic fibrosis or GIFT-CF studies. So the GIFT study is now in its third iteration. So on the next couple of slides, I'll give you a brief summary of the two previous studies. The first study began approximately 3 years ago with GIFT-1. The first GIFT study, GIFT-1, was a case control observational study that used MRI to compare postprandial changes in the gut in people with CF and controls. The aim was to see if we could identify differences in the gastrointestinal function between people with CF and controls using MRI. The second study, the GIFT-2 study, was a blinded, randomized crossover trial where we compared tezacaftor-ivacaftor to placebo, measuring the same MRI parameters in GIFT-1. The aim was to identify whether MRI parameters, and therefore gut function, changed when participants were taking tezacaftor-ivacaftor by comparison to placebo. So this slide shows flowcharts for both GIFT-1 and 2. On the left-hand side, in GIFT-1, 24 participants in total were recruited, the cohorts comprising of 12 people with CF and 12 controls, and all 24 were able to complete the MRI study. So the MRI study involved a series of 11 MRI scans over the course of one day, over a roughly 6-hour period, with 2 standardized meals being given, one at the start of the study and one approximately 4 hours after the first meal. On the right-hand side, in GIFT2, 12 people with CF were recruited and randomized to receiving tezacaftor, ivacaftor, or placebo for 28 days. Then they stopped taking the medications, or placebo, for 28 days, and then crossed over into the other arm. Sorry. So this slide summarizes some of the significant differences in the gastrointestinal function we found in the GIFT1 study. So we found that the oral cecal transit time, denoted by the stopwatches here, and this is the time it takes for food to pass from the mouth to the beginning of the large bowel, was prolonged in people with CF at 330 minutes. We also found that people with CF had more water in the small bowel, seen in the graph on the right, and that between time points 240 minutes and 300 minutes, circled here in blue, there was a smaller fall in small bowel water content post-second meal. This reflects diminished gastroileal reflex, where the ileum empties contents into the cecum in response to food to make space. We have termed this measurement delta TI. The results of the GIFT2 study are currently being written up for publication, but no differences were found between tezacaftor, ivacaftor, and placebo. To confirm these findings, we aimed to perform a study using more effective modulators on a larger sample of participants and given the modulators for a longer period of time. This has led to the GIFT3 study. So GIFT3 is a prospective observational cohort study comparing GI function using MRI before and after Kaftrio. 24 participants underwent 3 scan days using the same MRI protocol as before. The first scan day was before starting Kaftrio, and the second at 3 months, and the third at 6 months post starting Kaftrio. On this slide, I can show you some preliminary results from the GIFT3 study. We have found that post-Kaftrio, the small bowel water content of participants increases. So baseline, you can see here in the orangey red color, and then 3 and 6 months in the blue and the green color. The delta TI measurement also increased, showing an improvement in gastroileal reflex. To show that, this increase in delta TI is illustrated more clearly in this graph. We found that the increase in delta TI between baseline and 3 months and baseline and 6 months was statistically significant. To contextualize the size of the increase, we found that while on Kaftrio, people with CF had a delta TI measurement that was comparable to that measured in healthy controls in GIFT1. So moving on to what we are doing at the moment. So the GIFT3 study continues following further funding, allowing for participants, the same participants from before, to return for a further set of scans at 18 months post starting Kaftrio. And at the moment, we are also conducting a feasibility study using a shortened version of the MRI protocol in children aged 6 to 11 years old. So this body of work is a huge team effort, and I'd like to thank all of our collaborators and our funders. I'd also like to make a special mention to my predecessor, Dr. Christabella NG, and to my supervisor and principal investigator for the study, Professor Alan Smith, whose dedication and hard work has driven the GIFT studies. I would like to say thank you much for listening.
Well, thank you very much, and it'd be great if the panel could all maybe put their cameras on. Excellent. Well, thank you very much, everybody, for joining us. It's always difficult, but if you could possibly book any questions in the chat. I've just got a few questions I'd just like to ask the panel, and I'll just go to one of you, one after the other. So the real question is, the introduction of Kaftrio, has it actually changed the direction of your research? And how does your research impact on that 10% you can't actually get triple therapy? And actually also at this meeting, has that changed anything in terms of how you'd like to engage with other collaborators? So why don't we get to Alex, since you were the last speaker?
Okay, so in terms of the first question, whether the Kaftrio is changing the direction of our research, I'd very much say yes, in 2 ways. The first one being we are wanting to try and explain sort of the differences that we've seen in terms of small bowel water content following Kaftrio. So we're trying to sort of design studies to try and figure out where that water has come from. So is it from pancreatic secretions, or is it just other kind of changes? And also it's changed sort of the course of our MRI studies, in that because the Kaftrio has been licensed in the 6 to 11-year-olds, we recognise that to try and capture people pre and post Kaftrio, we're going to have to start looking at the younger age groups, those 6-year-olds. Hence why we've started a feasibility study in that group. And I'm a paediatric doctor by background, so I find that challenge very exciting moving forward, to be working with younger children again. In terms of how the research may affect those unable to benefit from Kaftrio, so we would hope that through the research, if we can gain a better understanding of the pathophysiology, being measured through MRI, we could hopefully help either ourselves or other centres to conduct research to try and create more tailored regimens outside of just CFTR modulators, to try and figure out what other medications are out there that may work, or allow other sources to try other medications that have not been used before. So hopefully, I mean, that's a lot further down the line, but I suppose that would be the ultimate aim from where we are at the moment. And yeah, in terms of other collaborations, I'm really excited to say that, you know, we at the moment work quite closely with Manchester. I've collaborated with Ryan in the past, and I know that as a centre, University of Nottingham will be moving forward to work quite closely with Leeds in the near future as well. So always excited to have sort of like new collaboration and sharing of ideas and things like that as well.
Well, wonderful, wonderful. And in fact, I just wonder, Alex, whether Jennifer's NHE3 drug might actually be really interesting in looking at the MRI. So, so I'm going to I'll move straight over to Jennifer.
Okay, thank you. So whether Kaftrio will direct our research, probably not specifically in our lab, but some of the collaborators that we worked on this SRC with have recently published research looking at the effect of Kaftrio on mini-guts, on organoids, and showing that it did restore the chloride and bicarbonate transport. That has really, you know, implications for future work. So how our research could affect those unable to benefit from Kaftrio? Well, obviously, because the aim of our project is to target NHE3, so we're CFTR independent. So this would mean that if it was shown to work in clinical trials, that everybody would be able to use it regardless of the CFTR mutation. So that's a really exciting prospect. So new collaborations. So our work has led to new collaborations being formed between some of the SRC co-investigators, and you know, stuff that I've heard today makes the I think the link between the gut and the lung a really interesting area to research more, whether it's the immunological effect or actual transfer of bacteria between the two sites. So there's, yeah, I think that's a real area to investigate, and it has a lot of potential for manipulation of the gut microbiome to kind of improve other symptoms. So yeah, I think that's it.
Wonderful. I'm going to move on to Ryan. You're already collaborating with Alex, but it'd be great if you could answer those 3 questions. And in the interim, anybody who's got questions, please get them ready or send them in the chat.
Yeah, so as you touched on, yeah, obviously Nottingham and Manchester have got the gifts of collaboration that's going on. I think how has Kaftrio changed our research? Well, I think it will change it in the sense of, okay, similar to the other modulators that we've looked at, the pipeline will still be the same, but the extended sampling license for 18 months, I think will be really interesting. I mean, in terms of what's out there in the literature at the minute, a lot of studies, well, there's not many studies that go that far. So it'd be actually interesting to see what the long-term impact of modulation is. Does it actually does it do a better job than previous modulators at restoring that sort of signature, community signature? And if it doesn't, that's when we need to stop by potentially looking at these sort of inhibitory factors, so antibiotics, diet, lifestyle. But on the flip side, it'll also be interesting to see how they change following long-term treatment with modulators, and if that is the reason why things change. In terms of collaboration, I mean, for me personally, diet's not my strong point. I know Laura's got a great foundation in nutrition, diet, so that'd be great going forward if we could get, like I say, a lot of my stuff or our stuff's been pilot studies so far. So if you could deep down into it and get bigger cohorts and really look at these this multifactorial nature of it, that'd be great. And I think that ties into the question as well with, obviously it's a shame that there's still 10% or so of people that won't be able to access the drugs. But in looking at the drugs, we are still understanding more about these sort of other co-founding variables like diet, like exercise, and that will still shed more knowledge on these areas that can be managed by patients who aren't on the drugs as of yet. So it is looking promising moving forward.
That's fantastic, Ryan. You better know that Laura's got 100 blood samples and stool samples matched
Fantastic.
in our freezers. All right, Ryan, that's fantastic. So Laura, we'll come to you and then we'll open up to questions. So Laura, do you want to answer those 3 questions?
Yeah, I'd love to. Thank you. So I can answer sort of the first and second one about how cafeteria affect our study and those without. In sort of actually the biggest impact was COVID, and that sort of tied in. So we planned the study before we knew cafeteria was going to be approved, and we planned it as an observational cohort study with 2 time points. And very roughly about half our study participants, we finished sort of did their baseline and follow-up samples before COVID, and then COVID came and we went on pause, and that's when cafeteria got approved, and then we reopened to collect the follow-up samples. So half our follow-up samples, by that time point, quite a few participants had started cafeteria. So we saw that as an amazing opportunity that we had data before we even of people's diets and their abdominal symptoms before we even knew cafeteria was coming in. So we have that. So we had that data, and we've shifted, and we made sure we are now looking pre and post cafeteria. But in terms of those who aren't eligible, the beautiful thing about our cohort studies, we're going to have data on those participants, and we're really keen, and I think it's really important there isn't much literature on how diet is impacting the gut microbiota, or or diet and antibiotics on the gut microbiota in such a large sample size. So we're going to very much be looking not only at how cafeteria has changed things, but also even pre-cafeteria to have that underpinning mechanistic insight into what it is. And then the third question was collaboration. I think, well, I'll just echo what we've all discussed. I think it's beautiful that we do have that variety. So our study has the advantage. So we recruited 175 participants. So we have a large number, lots of stool samples, lots of dietary data, abdominal symptoms, which is obviously fantastic. But we couldn't do that, and we wouldn't have had that sample size if we had mucosal biopsies, if we'd done MRIs. So you have that real depth of information, and I think it's beautiful, hopefully forming collaborations forward that we've got that different oversight and also different age groups. I think it's really important with our findings that we were very much in adults. Our median age was just over 30, so very different picture in terms of how CF and inflammation and what's reversible and what's not at different stages. So I think they will feed in, as very much said. Hi, everyone.
Brilliant. Thank you very much, all of you. So I think we're going to open up now for questions. Did you have a question? I saw a hand up that went down. No. Laura, do you want to just look at the chat, and you'll see that Gary sent you a very interesting question showing, I don't know if you want to speak, Gary?
Okay, thank you. I was extremely impressed by the data that you showed, Laura, because there's such a clear separation between CF, colorectal cancer, and which was basically overlapped with normal. And I think other speakers, the difficulty with having so many speakers is that they have actually touched on that this could all be due to antibiotic usage. But of course, nowadays, if you were to test a younger age group, and I think you've also touched on that, you may find that we're actually using less antibiotics as people are better. They may be closer to the normal, but would it be worth looking at a younger population and looking at those on lots of antibiotics and those are not, to see whether it is the antibiotic question?
Laura, do you want to answer that?
Sorry, I couldn't find the unmute button. Despite, so I think that's a really interesting question. I guess there are 2 separate sort of considerations. I think it is a changing picture. I think we have looked at sort of within that cohort, I did look at whether there was a separation between those CF samples in the orange, if we could see any patterns with those who had a high number of IV antibiotics course in the preceding year compared to those with a low. There wasn't a clear pattern. They weren't clearly clustering vision by that. So I think, but they're adults by that time point. So it doesn't appear acutely that it was the IV antibiotics driving any difference within the CF cluster. And I think we recently have done a systematic review on the gut microbiota in adults with CF, not specifically linking to colorectal cancer. And I think I don't know, but my personal view is antibiotics in the gut microbiota in CF is probably a little bit different to how antibiotics affect the gut microbiota in people without CF, because I think the primary CFTR defect, and I think when we look at mouse models, that fundamentally drives a different gut microbiota. And the additive effect of antibiotics on top of that, I guess, is the question. And some reviews have shown effects, and some have, when they've excluded those not on antibiotics, they haven't found as big effects. So I think that's something we need to investigate further. I suspect they do have an impact, but I suspect their impact wouldn't necessarily be directly comparable to an individual without CF on antibiotics. I don't know if anyone else has any other thoughts, but that's, as a PhD student, my current, not analysed all the data yet, and we will get the gut microbiome profiles, and for that we will do more specific looking at it. So this is very preliminary thoughts, if anyone had any.
Yeah, so Laura, I mean, one of the studies you did in the beginning was to look at the national database to look at rate of decline in lung function. And within the study, you've got high decliners versus low decliners. And I think that will certainly give it power. But I totally agree. Once we've isolated the issue and problem, I think it'd be really interesting to go retrospectively into pediatrics and over time and see how it is. So, you know, that could be a really interesting collaboration. So are there any other questions that people want to ask?
Yes, I have a question to ask. Can you hear me?
I can hear you beautifully.
Yes, yes. So my question is from Ryan. You're using some fantastic technologies to measure various things, but I worry about measuring short-chain fatty acid in the fecal samples, because that is not indicative of what is in the intestine. In the intestine, short-chain fatty acids are produced, but they're absorbed, and what is not absorbed, it goes into the feces. So if there is any problem with transporters that transport short-chain fatty acid, obviously if they absorb more, you have less in the feces. If you absorb less, you'll have more in the feces. So this is something you have to be very careful.
Yeah, no, I completely agree. I think on top of that as well, short-chain fatty acids in general are the storage of them is super, super important. Like you say, there's many ways that your analysis or interpretation can be skewed. So by just the basic physiology, but also by storage. So I guess considering that, that's why we're trying to tie in the predicted functionality as well. So if we have the predicted and what we're seeing, and also try and integrate to, you know, both the NMR, which is less sensitive, and the GCMS, which obviously is more sensitive, there's obviously the question of, okay, what can we see relative to each other? Obviously the physiology being impactful, but actually, you know, do we need to go down to the level of GC? Is NMR a good proxy, although it's not perfect? But yeah, no, I completely agree, and it's definitely a limitation, but it's definitely something worth investigating. And hopefully, yeah, if it's relative, it shouldn't be too bad. But yeah, completely agree that there are many limitations around it.
Thank you.
So just asking you, because you asked that question, how should we be measuring short-chain fatty acids, do you think?
Well, it's obviously the access in animal models, you can do it. There's no problem because you can take the content and you can measure to see how much it is there. In humans, it's going to be difficult to access the internal. And of course, the biopsies that they remove is washed. I think in humans, it's going to be very difficult. But if you're using animal models, you can take the content and measure. the gut content. And that applies also to microbiota. I always worry about looking at microbiota in the fecal samples rather than the content. And what we have done, and Jennifer explained, that we took the biopsies and we look at the mucosa attached to microbiota, which is much more close to the realistic, situation. The short-chain fatty acid, we just have to be aware that what is in the feces, it doesn't indicate what is in the intestine. And thank you for your reply.
Yeah, thank you very much. Who's asking any questions that I can see at the moment, but please do ask questions if you want. I'm going to go to Alex. Alex, I mean, MRI in your studies, I think it's 6 hours, isn't it, that the children have to be there. Do you think MRI will really take off in terms of analysing the gut, or is it just going to be a very specific tool just purely for research? And are there any techniques that you think will be useful?
I certainly think that's a good question, and it certainly challenges that as a group we have thought about, because yes, the study protocol is very intensive. We would require a participant to be there on the adult side for 8 hours, of which roughly 6 hours of that time would be in and out of an MRI scanner. For the kids, we are trialing it so that they only have to be in the scanner for 3 sets of scans, totaling about an hour to an hour and a half time total in the scanner. We're very fortunate in Nottingham that we have nice resources to try and make it as pleasant an environment as possible. But it's certainly a challenge that we recognise in order to lots of challenges that have to be overcome if we were to expand it and roll it out beyond sort of like the Sir Peter Mansfield Centre in Nottingham. Again, these are discussions we're having amongst our group at the moment to see how we can move forward, but we're hoping that as a first step, if we can show that the shortened 3-scan protocol can find the results we're looking for, at least then that's a first step in trying to see how we can make it even shorter. So we're really at the very beginning of that, but we're hopefully taking that next step with this feasibility study. I hope that answers the question.
That's brilliant. I'm going to be cruel to Laura now, and I'm going to say to her, do you think we need to modify the diet, and do you think probiotics are useful?
I think so. Before I give my answer as a dietitian, I have to say, I think every individual with CF really is an individual. So I would advise that they contact their dietitian for their specific needs. So I can only give an answer on a sort of more broad view. I think CF has historically been associated with undernutrition quite profoundly, and because of the intensive and hard work of the individuals with CF themselves and dietitians and the whole medical team, nutrition has improved, but it's improved to the extent that now average BMIs are holding pretty well, and now with cafeteria we're seeing that big weight gain, and lifespan is increasing, and we're seeing more of which is an absolutely fantastic, but we're actually seeing the complications, with the increased risk of colorectal cancer, cardiovascular disease, increasingly overweight and obesity, and outside of CF they are lifestyle and diet modified, and I think we are going to look at shifting the diet, and particularly I'm obviously interested from a gut microbiota point of view, whether that could be beneficial, because traditionally a high fat, low fiber diet often consumed by people with CF isn't optimal normally for gut microbiota health. So I think I'm hoping our data, when we get it, diet and the gut microbiota, will give us insight whether there is scope to really, I guess, living well with CF through modifying the diet, both in the short term but also in the long term.
That's Laura, that's fantastic. I'm going to cut you short there because we've literally got less than 1 minute, because I want to say that I think all 4 of you have been absolutely fantastic. I think the future of CF gut research is safe in your hands. I think the 4 of you and your supervisors should get together and collaborate, and Jennifer is going to help us with AI. And I'd just like to thank the CF Trust for their fantastic support and raising money to do this sort of research. So I'd like to thank you all. I'd like to thank all the participants for joining us, and have an enjoyable rest of the day. I see a hand just went up there. Okay, well, thank you very much, everybody. Thanks a lot.
Thank you very much.
Bye bye. Thank you.
Bye.